Welcome to Deep Down, the introductory talk to plant tissue modeling and plant root modeling, which is part of the Plant Cell to Plant Fields e-session of the CSDC 2015 e-conference. This introductory talk is divided into three parts. The first one, I'll try to highlight maybe how plants and plants tissue can be considered as interesting complex systems. In the second part, we'll be talking about how to actually model plant tissues. And in the third part, we'll be discussing about virtual root tissues and what we can do with those. So, without further ado, let's start with chapter 1. What actually makes plants and plant tissue interesting complex systems? Well, we could try to lower information on that. Compared to animals, plants are sessile organisms and they lack any kind of nervous system and any kind of apparent sensory organs. So they may appear a bit simple at the first glance, but we could basically abstract them as spatially constrained systems with no central coordination and only local limited perception. But they also exhibit very limited embryonic development, which means that a plant, while it's in the seed, has a very small root and a very small shoot and much more. And unlike animals, the main bulk of their development is actually post-embryonic, which means that they actually develop extensively through iterative secondary organogenesis. So that means that these organisms, which appeared at a glance quite simple because it was spatially constrained, it had no central coordination and only local perception, actually exhibit a very interesting property, which is a constantly growing structure. And this basically makes it kind of analogous with other bound, coordinated, growing, man-made maybe entities such as the internet, for example. And there is, from a theoretical point of view, a very strong incentive to study analog systems, to understand them better. So it may be worth it actually to look a little bit more into detail at plant systems. Okay, let's say for a moment that we'll study the behavior of the system. Imagine for one moment that you actually are a plant. You are a sessile organism. You have limited sensing capacities and perceive only local variation in environmental parameters, such as gradients of water, for example, in the soil. And you have, however, unlimited growth potential, provided that you can get the material for it. So, how do you grow? How do you make the most of your potential? Well, you could try growing everywhere at once, but exploring all space is actually a very poor strategy. And you're much better to rationalize and optimize your benefit-cost growth ratio to be evolutionary competitive. Much better off like that. And the key term here is optimize. Why? Because it's a theoretical term, and we can basically consider from a theoretical point of view, plants are optimization problem solver, which is a kind of interesting thing. For example, the IR parts are constantly trying to optimize light interception. Meanwhile, the plant roots, which grow underground, are constantly trying to optimize a problem of foraging through an heterogeneous media, and at the same time trying to distribute mechanical tension within the soil to ensure that the root keep the plant in place. So basically, as a plant, if we go back to that, how do you actually can optimize your growth? The growth being taken as the largest sense possible, meaning growth and differentiation. So how do you actually optimize your growth? Well, what you need to be is to be able to direct your growth and differentiation precisely. And that suppose that you need to be able to read and transduce environmental inputs into growth and or differentiation indications. Remember, you have no nervous system, no brain, no nerves, so you cannot send direct information and synchronize cell activities. So you need an alternative solution. And plants have evolved to transduce the outer information as biochemical and or biomechanical inner information fields. What do I mean? I mean that the cells will read information fields, those biochemical and biomechanical information fields, and react accordingly. For example, redirecting the growth in the direction of maximum light or maximum water content. So let's take an example to make it a bit more concrete, and let's talk a little bit about gravitropisms, the perception of a gravity field. Now, gravity is a very abstract concept. You can touch it, you can't smell it, you can taste it, but it acts on you nonetheless, and it actually acts on plants as well. And plants do perceive gravity. If you take a plant and you put it inside, what you'll see is that rather rapidly the plant will reorient its growth toward the new gravity vector. And what's interesting for us is to try and look at what's happening in a little bit more detail during this transition process. So let's look at the root tip, the very root tip. If you look under a microscope and you use different kind of molecular marker, what you could put into evidence is that there's actually a constant active reflux of an hormone which is called auxin, and this hormone is flowing down the center of the root. It's got to the tip, move to the side, and move back up right up the side of the roots. So this auxin, it may be important to say that it impacts lots and lots of cellular responses, including cell growth. Auxin is actually a key player in the information field in plants. Let's go back. We've moved our plant onto the side, the gravity vector is reoriented, and what's happening at the tissue scale? Well, what's happening is that you actually see some reorientation of the active transport of auxin, which will direct more auxin toward the bottom of the root, the new bottom, and which will end up by generating a new pattern of auxin distribution with more auxin content on the bottom. And as I said, auxin actually impacts cell growth. So what this will ultimately lead to is that the asymmetrical distribution pattern induces a differential local growth with longer cells on the top, shorter cells on the bottom, and that means basically a reorientation of the tissue. So, taken all together, in this very specific case, the information regarding the orientation of the gravity field is transduced as an hormonal field whose pattern the cells can read and interpret locally. So, as I've said, plants can be perceived from a theoretical point of view as optimization biological machines, and they integrate constantly multiple information fields during their development. And the crucial step is that these information is transduced efficiently at the cellular level. And these transition processes are ultimately cellular patterning games. What I mean by that is that each cell within the plant has a copy of the instruction, the actual genetic code, which is needed to interpret the local information field that the plant has generated and to eventually change it. Though the existence of constant feedback loops between the structure, which is a tissue, and the overlaid information field is the source of plant systems' complex behavior at the cellular and whole plant scales. What do I mean by that? Well, if you take a plant which has generated an inner information field, this field of information will affect the cells, which in turn will affect the field, changing it, and you basically circle on and on between the information field and the tissues. And those looping behavior are way too complex to be apprehended with classical approaches, which is why to perfectly understand and perfectly predict such complex behavior, new approaches were needed. And this is why biologists, mathematicians, and computer scientists are currently focusing on research effort and modeling efforts to create virtual tissues to help solve the patterning game in silico. And this brings me to my second chapter Now that we've seen that plant and plant tissue are actually quite complex, how can we actually now solve those complex systems with the help of some models? Okay, so let's try and load up some models, some tissue models. I guess we have some definition work to do first. So that's a personal experience that I'll be writing now. And what I've observed is that when biologists meet with computer scientists to design virtual tissues, the very first discussion often runs more or less like this. Okay, so we want you to help us make a computer model of this tissue. Well, I'd like very much to, but what do you exactly mean by tissue? What exactly is a biological tissue? It's a very good question, actually. And by formation, biologists usually take the answer to that question for granted. But let's take a moment to see it from both the biologic and the computational perspective. Try and understand why the computer scientist may ask this question. So what exactly is a tissue? Well, the intermediate scale between the whole organism and the molecular network, and basically a cellular network. That's the basic answer for a biologist, and it's a very correct definition. But having heard this kind of answer before, I'd get that the computer scientist would go, no, you've misunderstood. What I actually mean is what exactly is a tissue. Kind of interesting, isn't it? Can you tell me how the cells are connected? Are they touching? How are they touching? Can you tell me if the tissue that you're looking at is 2D or 3D? Do you look at the time or just at a single moment in time? Are the cells moving, dividing? What are the rules that govern those movements or those divisions? What's the time scale for the cell dynamics? How are cells communicating with each other directly, indirectly? Do you consider genes networking within the cells? Or how would you describe the cell inner structure? Let's stop there. And let's spare the poor biologist who is slowly getting tunnel vision from this avalanche of questions. And let's take a little bit of time to wonder why is the computer scientist asking all those questions about a simple tissue. Well, it turns out actually that all those questions are related to computational choices which may change the way the model is built. Let's go over them one by one. First, can you tell me how the cells are connected? Is a matter of topology, which means the actual relationship, the description of the relationship between the element in the system. And there are multiple computational ways to represent a cell network. So you have to make a choice here. Is tissue 2D or 3D? It's a matter of geometry. Well, geometry itself is not a matter overly complicated, but you have to take it into perspective with the other computational topics. 3D is way more computationally complex than 2D when it is related to other topics such as mechanics. How are cells moving, dividing, how? You have multiple ways to implement physical cohesion growth, for example using springs or tensors. It's much more difficult in 3D than in 2D. You have also to take into account a description of the tissue viscosity, meaning the way the tissue will deform when you support some kind of constraint. And there's also multiple ways to divide cells, and you also have need to keep the tissue as a computationally tractable data structure, meaning that you need to be able to track all the changes in neighborhood between cells due to cell division and cell movement. And this is can be difficult in 2D, and it's much more difficult in 3D. So this brings us actually to the data structure. How would you describe the cell inner structure? Is a question that would directly relate to the way you describe your tissue, your cell network. Do you consider that cells are single nodes? Or do you consider that cells are groups of nodes? Or do you actually consider coupled network with interplay between different networks at different scale? It's all a matter of data structure choice here. Now, how are cells communicating with each other in the matter of signal propagation? Is there any kind of contact mechanisms? Are active and passive transfers of single molecule needed? Do you need to implement physiological rules? All that fall under this topic. What's the timescale for the cell dynamics? Is a topic of time integration. Now, time integration sounds rather simple, but actually when you have to mathematically integrate very short time steps and very long time steps or time scales, it can get very fast complicated, and it can drive you to having some kind of divergence issue with the mathematics. So, last question, do you consider gene networks in the cells? Is related to the topic of multi-scale modeling. Basically, what the computer scientist here is asking is whether you have to add a model within a model, having a network which defines for all kind of genes or molecule their behavior on the cellular level, and having the cells themselves exchanging with each other, exchanging signal, reacting to each other. So basically the whole multi-scale deal. Alright, so even before starting to build a model, you may have to consider all those fundamental computational issues, and which means that tissue models are very much a matter of viewpoint. And you have to ask, what is your favorite biological question? Or think in another way, which information field are you actually interested in? And how will you present it and consider it for all those reasons? So, for example, are you interested in the information within hormonal fluxes at a small time scale? In which case you basically need, I guess, some topological description, a geometric description, some kind of signal propagation mechanism, and an adequate data structure to keep track of all that. Or maybe you would be interested in the cell growth field and tissue mechanics at a large time scale, in which case you'd need topology, geometry, a little bit of mechanics and time integration, and of course the adequate data structure for all that. Now maybe you are interested in the complete feedback loops of the environmental optimization pattern in games, in which case good luck, you may need it, because you may very well have to take into account everything that I've been talking about from the computational point of view. But wait, there's more, always more. We have only talked about the implementation of the model itself and not what comes before or after modeling. What do I mean by that? Well, for example, do you want a faithful representation of your tissue coming from picture, maybe? Well, that means that you'll have to tackle the image analysis issues. Being able to automatically or semi-automatically or manually convert an image in a data structure is not an evident process. Or maybe you would like to actually see the output of your model and interact with it, being able to click on it and change its property on the fly. Well, I guess then you have to work on the visualization and graphical user interface issues, which are also a very tricky topic and time-consuming topic as well. So, in summary, tissue models behave a bit like this. They need to feed on biologists, or at least on biological data of all sorts. And before asking any question about your information field of interest, you first have to feed the model. And now that we have a pretty good idea of the extent of data that the computer scientist needs for their model, can we actually have enough from the biological point of view to meet with those demands? Well, fortunately, yes. We can rely on all the basics of cellular and molecular biology for animal cells or plant cells. We basically know a lot about cell growth, cell division, differentiation, about hormones and molecular networks. It's actually rather more a matter of choosing where to put the feeding limit for the model. Let's make also a little bit of a quick parenthesis here. All that I've been saying from now on tissue model applies equally to plant and animal tissues, because the computational principles behind those are common. So the main difference between animal and plant tissue models will lie in the mechanical principle and tissue plasticity. Animal cells can grow, divide and move, while plant cells are enclosed in the cell walls and can only grow and divide. This actually implies that plant tissues are actually much more simple to consider from the topological, geometrical and mechanical point of view because there is less cell movement, no changes in neighborhoods. And this is definite advantages of plant tissue versus animal tissue from the modeling point of view. Things are complex enough already. See, all the plant tissue agrees? Hello, are you plant cells? Okay. Let's now say we have fed our model with enough biological data and have chosen a question of interest. The computer scientist has finally called it a root tissue model. But what does it look like? And this brings us to our third chapter, the virtual root tissues. We talked a little bit about generic tissues, and root tissue models are mainly based on Arabidopsis thaliana. This model plant has a lot of advantages. It has a very simple root tissue structure, and its roots are easily accessible, and there is extensive available genetic and molecular resources to study it. It's a very good model for organogenesis. So let's have a look at what makes a root at the cellular level and at some way we can model it. Here's an Arabidopsis root tip. It's where most of the tissue patterning takes place, which explains why it's the most frequently modeled root tissue. Simple tissue architecture. It's made of concentric cell cylinders which taper to a point at the root tip, and this root tip is actually responsible for environmental perception processes. If you go up in the root, you'll encounter the meristematic zone where the cells divide and start differentiating, and if you go even further, you'll end up in the elongation zone where the cells are growing. So basically here you see that the root tissue exhibits a longitudinal donation pattern. And remember what we say about pattern and tissue scale. They come from information field, which means that there is a kind of longitudinal, quasi-one-dimensional information field at work here. And this field is directly responsible for the control and global roots control of global root growth through local cell division and elongation. So can we actually investigate this information field using root tissue model based on the principle we've described earlier? Well, we can. We could go at it from the biomechanical point of view and focus on the elongation zone. That's, for example, the work of Fosler et al. In 2013, and they have described a pure biomechanical model based on relative geometry and with a gradient of auxin, which drive the cell elongation. Or we could also go at it from a biochemical point of view and focus more on the apical meristem. And that may, for example, the work from Mironova et al. 2012, with a simple biochemical model of hormonal fluxes, which is based upon an abstract static tissue with a regular lattice of square cells. Now we can also go way further and describe a complex genetic network within a dynamic abstract tissue, and that the work from Marlin et al. 2014 we described an extensive genetic networks and all kind of information fields which are hormonal gradients and rules for cell division, cell growth, and all kind of things. And basically, we're able to recreate the donation pattern we have mentioned earlier. So that's three completely different models and three completely different ways to consider root tip patterning dynamics, but they are all equally valid. It's a matter of point of view. And let's have a look at root from another point of view, actually. And let's look at it in section. We said earlier that the root is made of a set of concentric cell cylinders, and roots actually exhibit a regular pattern of differentiation within and around the vascular cylinder, the inner part of the root, which means that there's here again a bidimensional information field at play, which pattern the vascular tissue. And these patterns also, by incidence, pattern the position of potential root branching sites, which means that these very local bidimensional fields had a global impact on root system architecture as a whole, because it drives the branching process. So what can we say on this from a computational point of view? Well, basically what you could say is that you would describe it at a C-scale, and you would integrate also hormonal physiology and a gene network, and that the work of Herrera et al. 2014, and they were able to basically recreate the distribution of hormone and gene activity within the root tissue, meaning that they were able to capture in wild type and mutant the driving phenomenon behind the information field that are responsible for the patterning. All right, so we've looked at some longitudinal and radial root tissue models which are perfectly able to provide insight on some patterning events. But before moving on to my conclusion, I'm speaking entirely for myself. I'd like to say that those last models are somewhat lacking in challenge and impact. And I guess I'm not making any friend here. But why? Because they consider only bidimensional ongoing patterning events within already established tissue. And I guess that more interesting models would require more challenging processes, such as, for example, the initiation, development, and emergence of a new lateral root from within the root tissue. This process of lateral development is a complex patterning process which is akin to divine embryonic development, which starts from a very few cells and move toward its ultimate fate of being a three-dimensional differentiated organ. And the lateral development is a three-dimensional interplay of information field and feedback loop, which is repeating itself from scratch over and over and over again during plant development. Now, this is actually a patterning process worthy of modern research, and so much so that it's a process which has yet to be properly described, fully described, and let alone be modeled. So it's kind of the cutting edge for plant root models. So to conclude my talk, I'll just summarize a little bit what we've seen. We've talked about the fact that plants can be considered optimization problem solvers, about the fact that tissue pattern arrives from information transduction and feedback at the cell level, about how computational tissue models are all about biological question and the definition of an information field of interest, and how root tissue models can be used to investigate different geometries of patterning information fields, and also how lateral patterning in development is still elude modeling effort. So I guess it's no mystery that I would basically base my perspective on these last points and kind of try to answer the question, what is still missing to be able to tackle this lateral patterning issue? Well, I'd say 3 points are missing. First, we're lacking an in-depth 4D geometrical description and 3D plus time of the tissue physiological state. Then we're lacking also a precise understanding of the biomechanics at the cellular level. And finally, we're lacking a comprehensive concept for global gene networks interplay in a developing organ. And if we were able to bring all those 3 points together, I guess we'd have enough to actually build up complete virtual roots. So let's end up here my introductory talk to plant root tissue models. I hope you enjoyed it. If you have any question, feel free to add them in the e-room session. And of course, I'd like to acknowledge also all the people who've made this work possible, and thank you for your attention.